Hello, my name is Nicole Mullen, and I'm an undergraduate researcher in the Guillemin lab at the University of Oregon in the Institute of Molecular Biology. My research explores the role of the microbiota in the development of insulin-producing cells in the fruit fly, Drosophila melanogaster. So animals are complex systems of interacting host and microbial cells. The host-microbe relationship can have widespread effects on everything from obesity to cancer, inflammation, and significantly for my research, metabolic systems and growth and development. So figure 1 illustrates previous research showing that resident bacteria are required to increase the number of insulin-producing beta cells in the larval zebrafish and also in the mouse pancreas. A secreted bacterial protein called beta cell expansion factor A, seen in figure 2, or BefA, is sufficient for the expansion of this cell type. In flies, the equivalent insulin-producing cells, or IPCs, are located in the brain, with 7 IPCs located in each lobe, as seen in figure 3. My research worked to see if this relationship between microbiota and insulin-producing cells is conserved in fruit flies. Doing this has the potential to show a powerful relationship between metabolism and microbiota conserved across multiple model or organisms, and may have implications for the effect of the microbiota on the brain. I have 4 aims in this research. My first aim is to investigate if the microbiota is required for normal numbers of IPCs in flies. For my second aim, I tested if feeding larvae BefA protein could rescue IPC numbers like it rescued beta cell numbers in zebrafish. My third aim tested whether ectopically delivered BefA could rescue IPC numbers. And my fourth aim investigated the mechanism of BefA by testing whether transgenic expression of the TSL gene, a no-pore-forming protein, could also rescue IPC numbers. For my first 2 aims, I counted the number of insulin-producing cells in each brain lobe using fluorescent microscopy. In order to count the IPCs, I dissected, stained, and imaged third instar larvae expressing green fluorescent protein, or GFP, under the direction of DILP3. So when DILP3 is expressed, the IPCs glow green, as seen in figure 4. This allows me to count and compare IPC numbers in germ-free flies, where all of the microbes have been removed, versus conventional flies whose microbiota is left intact. For the feeding assay shown in figure 6, all flies were fed a liquid diet of sucrose and bromoethanol blue dye, as well as the BefA protein. Because the larvae are translucent, the blue dye was visible in their intestinal tracts and served as a check to ensure that the larvae ate the food. The experimental group of germ-free flies was fed the same liquid fly food with added BefA. The same quantification methods from the previous experiment were also used for this. For the ectopic expression of BefA and of TSL, we used the Gal4-UAS system from yeast to express these specific genes only in the DILP3 positive cells in flies. I then compared IPC numbers between conventional flies made germ-free and flies made germ-free with a BefA or TSL transgene insertion. For my results, in addressing aim 1, brain imaging and IPC counting demonstrates that germ-free flies have fewer IPCs than flies raised conventionally. Figure 7 shows this quantification, with experimental groups on the x-axis and the number of IPCs per lobe on the y-axis. The difference between groups is statistically significant by t-test. Figure 6 shows representative images of these conventional and germ-free fly brains. For this analysis, I quantified IPCs from 253 brain lobes. In addressing aim 2, the BefA feeding assay showed that feeding larvae BefA protein can partially rescue germ-free larvae, meaning that they have IPC counts closer to conventional flies. In figure 9, IPC counts for the 3 groups, conventional, germ-free, and germ-free BefA are shown. There's no difference between the germ-free and germ-free, or between the germ-free plus BefA and conventional groups, but there is a difference, though slight, between germ-free and germ-free plus BefA. While the germ-free flies still had fewer IPCs, germ-free flies supplemented with BefA protein had IPC development more similar to conventionally raised flies. Qualitatively, flies fed BefA were larger than the germ-free flies. For AIM-3, ectopic expression of BefA also showed partial rescue of IPCs in larval brains. In figure 10, you can see the recovery of the IPC counts from germ-free to near conventional levels. These results mirror those found in zebrafish and suggest that BefA is sufficient for the expansion of IPCs. For AIM-4, ectopic expression of TSL, a known pore-forming protein found naturally in the fruit fly, yielded interesting results. For a portion of the brain's image, there was the expression of ectopic DILP3 positive cells throughout the brain. This could suggest that the pore-forming mechanism of the TSL bacterial of the TSL protein is what allows IPC cells to develop normally. In conclusion, my research showed that microbiota are necessary for normal IPC numbers in fruit flies, as germ-free treatment led to a decreased level of IPCs. BefA protein alone, when delivered through a feeding assay or ectopically, is sufficient to partially rescue IPC numbers in germ-free flies, making them closer to conventional. And finally, expression of a powerful, naturally occurring and known pore-forming protein led to an incredible increase in the DILP3 positive cells throughout the brain, suggesting that IPC expression could be dependent on bacterial protein ability to permeabilize membranes. Future experiments will focus on the mechanism of BefA, examining the functionality of this bacterial protein in a developmental assay. So what does all of this research mean? Well, we know that the microbiota has a powerful effect on the metabolic pathway, fundamentally affects cell development, and can influence the brain. This information could help us treat diseases like diabetes, better understand growth and development, and have implications for microbiota's effect on the way that we think. Finally, I would like to thank the University of Oregon's SPUR program, the National Science Foundation, and all of the members of the Guillemin Lab for their support in this research. Thank you.